Welcome. Welcome to this session. This is the sanitation session, and we've got 4 presenters lined up. But we'll have our first presenter, and that will be Valerie, who will be talking to us about willingness to pay and the demand for sanitation products in rural northern Ghana, and looking at the impacts of seasonality and methodology. The floor is yours, Valerie.
Great. Thanks for the introduction. As Musa said, I'm Valerie Bauza, and I work for the Aquaya Institute. And he already gave you an introduction to what I'll be presenting. So I'll start with some background. So despite very considerable global progress in reducing open defecation, it still remains common in some areas, such as in Ghana, where the JMP estimated in their most recent report that 31% of the rural population still practice open defecation in 2024. And this remains a challenge, particularly in northern Ghana, where there are unstable soil conditions that often cause low-cost facilities that are made with low-quality materials, often as part of CLTS, to collapse or cave in each year during the rainy season, which leaves people to revert back to open defecation. And there's some pictures on the slide that I took last year when I was in Ghana of some examples of latrines that had caved in, and then the households had reverted back to open defecation. And so in order to get sustainable sanitation in northern Ghana, it requires the use of more expensive technologies that include these durable pit linings in order to be sustainable over time, particularly in these soil conditions. And that may require subsidies, especially for low-income households, to access them, because in this area of northern Ghana is also areas that has very high levels of poverty. So the objectives of our study were to determine prices at which rural households in northern Ghana were willing and able to pay for these contextually appropriate sanitation products, to identify factors that may affect a household's willingness to pay, and to compare and offer recommendations regarding different approaches for measuring willingness to pay. And I'll cover all of these objectives, but mostly focus on the last one. We looked at 4 different types of potential durable sanitation products for this work. And there's pictures of the different types on the slides, and I think most people are familiar with them. So just to briefly summarize, we looked at the Dingy-Loo slab, which is a plastic slab with plastic substructure rings to be used for the pit. We also looked at Sadopan toilet with concrete block substructure, as well as water closets that used flush toilets to a septic tank, or a biofill flush toilet to a biodigester. We used 2 different methods to determine willingness to pay. The first was looking at stated willingness to pay, which used a double-bounded dichotomized choice method of contingent evaluation. And we did this with a series of 3 questions. The first question asked the respondent if they would be willing to pay a certain amount for the toilet facility, and based on whether they responded yes or no, they would then be asked a similar question with a different price point. So if they said yes to the first question, the second question would be asking them if they were willing to pay a higher price, and if they said no, the second question would be asking them if they're willing to pay a lower price, and then the final question would ask what's the maximum amount your household is willing to pay. And this would be the number that we would use for demand curves and to understand average willingness to pay, and sort of this series of questions helps households think through kind of the thought process of what they're willing to pay. And the first number that they get for the first question is randomly generated among a range of price points that we expect to be reasonable price points in the area, because sometimes that first number can anchor a price in their mind. So we want to randomly vary that number so that we get a good sense of willingness to pay across a range of prices. We also looked at revealed willingness to pay using an auction method to see what households would actually pay for the sanitation technology. And for this, we used the Dingy-Loo slab that's pictured here and that I mentioned on the earlier slide. And in this example, households would place a bid of how much they would be willing to pay for the Diggy-Loo slab, and there were eight price points that were sealed in an envelope, and so the household would randomly select an envelope, and if their bid was greater than or equal to the price that was in the envelope, then they won the auction and purchased the slab for the price that was in the envelope, and if their bid was lower than the price, then they lost the auction and they didn't purchase the product. And so this was to understand how much they would really be willing to pay in actuality. We collected several rounds of data collection that are summarized on the slide. First, we did a baseline survey that looked at stated willingness to pay for all 4 of the toilet facilities, and then we followed that up with the sort of 2 different types of the revealed willingness to pay experiment, where we first visited households, collected their stated willingness to pay for the Diggy-Loo slab, and kind of introduced what the auction exercise would be so that they would be ready for us to come back a few weeks later and be able to kind of make the decision as a household, is this something we want to purchase? Can we set aside funds to make that purchase before coming back to do the auction for the revealed willingness to pay? And we did this in 2 different seasons. One in the rainy season when households are typically planting in this rural area of Ghana where there's a lot of agriculture, and once in the harvest season, that was the dry season, and when a lot of income for the households is generated from their crop harvest. Our first key finding was that when we looked at stated willingness to pay for those 4 durable sanitation facilities that I explained earlier, the willingness to pay that they described was far below the actual cost of those facilities. And on average, depending on which facility, it averaged between 11 to 31% of actual costs. And so this shows the demand curve for those facilities where the cost is on the x-axis, and the y-axis shows what percentage of respondents were willing to pay that cost. And then the dotted lines show the actual cost of the facilities. And you can kind of see in all 4 cases, very, very few respondents were willing to pay the total actual cost for those facilities. When we looked at the results from the revealed willingness to pay exercise across both seasons for the Diggy-Loo slab, we found that it was very similar results where essentially, most households weren't willing to pay the full cost, but that the revealed willingness to pay was always lower than the stated willingness to pay. So they had overestimated the amount that they thought they would be willing to pay for the product, but we saw that their stated willingness to pay was more accurate in the harvest season when they often had more income than in the non-harvest season when they didn't. And so overall, in the harvest season, the revealed willingness to pay was about 24% of the current market price for this product, which was 800 Ghana cedis, and that price was already a subsidized price of the total cost, so it would be about 14% of the total cost. That was sort of their average willingness to pay. But on this graph, there's 4 curves that represent either the revealed willingness to pay in both seasons or the stated willingness to pay. And the stated willingness to pay is the dotted lines. And you can kind of see for the blue, which is for the harvest seasons, the curves are closer together than for the red, which is the non-harvest seasons, which shows that the stated willingness to pay was better reflective of the actual willingness to pay from the auction exercise in the harvest season compared to the non-harvest season. And this sort of shows the same thing, maybe in a bit simpler way than the curves, but you can kind of see overall with, again, the same colors. The dark blue shows the revealed willingness to pay in the harvest season, and the lighter blue is the stated willingness to pay. And so you can see that those 2 bars are closer together than the 2 red bars, showing that the revealed willingness to pay was more represented by the stated willingness to pay in the harvest season than the non-harvest season, and that the overall actual ability to pay was a bit higher in the harvest season compared to the non-harvest season, where it averaged 191 Ghana cedis compared to 171 in the non-harvest season. This is one other way to sort of look at these same results, where we looked at the ratio of the revealed willingness to pay to the stated willingness to pay, and when that is less than 1, and on the left side of the graph, that shows that the revealed willingness to pay was less than that. And so in the non-harvest season, it was about 53% of what they stated they would be willing to pay with these survey questions. But in the harvest season, that improved a lot, and it was up to it was about average 77% of what they stated they would be willing to pay. So when you sort of put these findings together, and if you want to use willingness to pay to sort of determine a subsidy level, what you realize is that the method that you use for collecting willingness to pay data can have a big impact on what the recommended subsidy level would be. And so let's say we want to choose 75% of access to this sanitation technology as where we want to place the subsidy level. There's 4 points on this graph that are what the 75% willingness to pay level would be, depending on the 4 different methods that we use. And so the y-axis shows what percentage of population would actually be willing to sort of purchase it at different prices that we could choose the subsidy for, depending on what method we use. And it can range from what we were hoping would get 75% access might go all the way down to 35% if we use kind of the willingness to pay method that seemed to overestimate actual willingness to pay the most. And so finally, we used multivariate regression to look at what factors influence willingness to pay. And we found that location, gender, and wealth all influence willingness to pay in our study data. And so these are factors that could also be considered when looking at subsidies. And so to sort of summarize some of the key takeaways of the study, we found that willingness to pay levels were far below all sanitation products' actual costs, suggesting a need for subsidies. Households stated willingness to pay typically overestimated their revealed willingness to pay for this digging loose lab, especially during the non-harvest season, and stated willingness to pay was more consistent with revealed willingness to pay during the harvest season, which also corresponded to the dry season and when toilet construction was more likely in this context. So when possible, we recommend the use of revealed willingness to pay over stated willingness to pay when possible, but it's often much more expensive and may not always be feasible. So in those cases, our results suggest that stated willingness to pay is more likely to accurately reflect real market behavior if those studies occur at a time close to when participants have more funds available and are more likely to make a purchase. So thank you for listening to my presentation. A few months ago, we published a paper on this study. So if you're interested in more details about this study, I can only cover a small portion of it. Please scan the QR code and look into it in more detail. And I'm also happy to take any questions. Yeah, exactly. And so I think you want to try to market them and sell them at the time where people are most likely to have funds available. So in rural contexts, that might be in the harvest season when they have a lot of money from crops, but in other contexts, there could be other seasonal income. And so it's helpful to consider that in your marketing.
One more question. We have time for one more question. Thank you. We'll have our second presenter. So we'll have our second presenter, and that will be Chimdi from the University of North Carolina, who'll be talking about optimizing swine waste bioconversion with black soldier fly larvae to protect public health in North Carolina. Chimdi, the floor is yours.
Good afternoon, everyone. My name is Chimdi Muoghalu. I'm a PhD student with the Manga Lab at the University of North Carolina at Chapel Hill. Today, I'll be presenting on a project which we undertook in the fall of 2024 with the title, Optimizing Swine Waste Bioconversion with Black Soldier Fly Larvae to Protect Public Health in North Carolina. For this, I would start by giving a premise of the project, the methods, the results, implications that drive future research areas. North Carolina ranks as the third largest swine producer in the United States. As shown in the figure 1, you see that some counties produce have more than 100,000 heads of swine. While this sector is economically viable, it generates large quantities of waste. Estimates show that over 10 billion gallons of waste are generated annually in North Carolina. This waste is harmful as it contains organic matter, nutrients, pathogens, which when released into the environment without treatment can contaminate surrounding soil and water bodies. Conventionally used methods for treating these waste materials, which involve storing them in large outdoor lagoons, usually have issues ranging from odors, ineffective treatment, and so on. For this, we propose the use of black soldier fly larvae composting to treat the swine waste. This is because it's a biological treatment process. It's also aerobic in nature, and also the waste material can be valorized. The black soldier fly can be valorized. More importantly, black soldier fly larvae are voracious eaters and that they can digest in large quantities of waste within a short period of time. The ability to digest large quantities of waste is driven by their digestive enzyme activity. The digestive enzyme activity, in turn, is influenced by the gut microbiome, the group of microbial communities that are found in the gut of the black soldier fly larvae. The tricky thing is that the gut microbiome of black soldier fly larvae is not conserved, but rather this depends on the rearing conditions, feeding rate, larval density, moisture content, substrate microbial communities. In this study, we focused on moisture content, and the reason why we focused on moisture content are, first, the moisture content determines the texture of the swine waste or the material you're feeding the larvae. Black soldier fly larvae, they are crawlers and they have to wriggle through the waste to feed effectively. Another reason is that black soldier fly larvae composting is aerobic in nature, and so when you have so much moisture content, it brings anaerobic conditions. Now, where we try to link moisture content to digestive enzyme activities and gut microbiome, if you look at so many studies or studies in literature, there are limited studies linking this role of moisture content on gut microbiome and, in turn, their role in the digestive process. To systematically link the bioconversion cascade, the objectives of our study were, first, to determine the impact of moisture on the performance of black soldier fly larvae, looking at larval growth parameters and contaminant reduction. Second, to characterize the temporal microbial succession and drivers across different moisture content levels. And third, to identify mechanistic bioindicators for successful bioconversion. This study was mainly experimental and involved rearing black soldier fly larvae on swine waste in dark containers. It involved single-factor experiments having 3 levels: moisture content at 65% representing low moisture content at 75% representing medium moisture content levels, and moisture content at 85% representing high moisture content levels. The containers were set up in triplicates with controls, and we had 2 trial runs. To assess the effectiveness of the treatment process, for the black, we harvested both the larvae and the swine waste samples over time for a period of 10 days daily. And for the swine waste samples, we analyzed for different parameters, including total nitrogen and total phosphorus as proxies for nutrients, total organic carbon as a proxy for organic matter, as well as pathogenic, as well as E. coli, which is a pathogenic indicator. For the larval samples, after killing and dissecting them to extract the gut, we analyzed for larval protein, larval enzymatic activities, including alpha amylase and the larval protease activity. For samples which we got on day 0, 5, and 10, we analyzed for microbial communities for both the swine and the larvae samples. We started with DNA extraction, and diluted DNA was amplified, and we pulled all the barcodes together, which we used for sequencing, which was done in Oxford Nanopore Minion sequencing device. The sequencing was done in MinNose software on ONT Cloud, and taxonomic classification was carried out using the 16S workflow tool. While Epi2Me helps us to be able to carry out taxonomic classification, it does not help with statistical comparison across different larval samples or different substrates. And so we had to import the data into R for data wrangling and some microbial analysis. Result. For larval growth, we observed that larvae read at 65% moisture content was more effective, looking at the larval weight, looking at the protein content, and the protease activity. For contaminant reduction, where we looked at total organic carbon, E. coli, total nitrogen, and total phosphorus, we observed that generally for all the larval samples and across all the treatment levels, the larvae was effective at removing E. coli and total organic carbon. But when it came to total nitrogen and the total phosphorus, it was not so effective. But when you look within the different treatment levels, between the different treatment levels, we observed that those read at low moisture content were more effective than those read at medium and high moisture content. Now, from larval growth and contaminants reduction, we can deduce that 65% moisture content was more effective for the treatment of the waste materials, that the larvae read at 65% moisture content were more effective for the treatment of the materials. Looking at the microbial communities, we looked at the phyla and the general levels. For the phyla levels, we did it so that we can see the overall microbial community structure. But at the general level, we did it so that we can identify functionally important microbial communities. The figure 11 shows the relative abundance of the larval samples over the 10-day periods from day 0, day 0, day 5, and day 10. And what we see that the dominant phyla there are Bacillota, Bacteriodota, Pseudomonadota, and Actimyceta, with Bacillota being the dominant microbial community. The reason why we have Bacillota as the dominant microbial community is that they are very common in waste materials as well as larval samples. In previous studies, we also observed the same thing. Previous studies also report the same thing. But what you can see is that as Bacillota reduces over time, if you notice from day 0 to day 10, there is a reduction. There's an increase in Bacillota, but there's a reduction in Pseudomonota. This is because Bacillota contains microbial communities that are fermentative and they are anaerobic. And as the black soldier fly is growing over time, you notice that its gut is getting more anaerobic, and that's why it's able to have more anaerobic microbial communities. But the Pseudomonota contains aerobic microbial communities. And so in the larvae samples, they will try as much as possible to have more advantage by having microbial communities that are anaerobic in nature. Conversely, for the swine waste, what we observed over time was that the Bacillotas reduced over time, while Pseudomonota increased over time. And the reason for this is that while composting is going on, the swine waste becomes aerobic in nature. And so while there are more spaces, there is more oxygen level, and that's why Pseudomonota is increasing over time. Now, we did this analysis to be able to identify enriched microbial communities. Since we observed that 65% moisture content is optimum, we decided to look at what microbial communities are enriched at the 65% moisture content, which are denoted by the red signs. What you see with this is that we see, for the general, we see families like Psychrobacillus, Peribacillus, or things like Psychrobacillus or Peribacillus. Now, these are microbial communities that have been linked to the production of enzymatic activities, hydraulic enzymes like alpha amylase and protease. And that could explain the reason why when you look at the 65% moisture content, they have higher enzymatic activity and they are more effective in breaking down organic matter. Also, we carried out principal component analysis to be able to link the microbial communities to the enzymatic activity. And from the ellipse of the principal component biplot, especially the one represented with red for 65% moisture content, what we see is that the protease points towards the 65% moisture content, showing that the microbial communities with the 65% moisture content are more effective at producing protease activity. Now, we've been able to link moisture content with gut microbiome and enzymatic activity. The big question is, what's the implication of this, especially when it comes to treating swine waste in North Carolina? So implications of this, first, we know that 65% moisture content, they influence the gut microbiome and in turn the digestive enzyme activity. But when you look at swine waste, especially those that are stored in lagoons and all of that, they have greater than 80% moisture content. And so we propose composting this with, because black soldier fly larvae is a composting process, composting it with waste materials that can help to increase the pore spaces within it, and at the same time can help to induce these microbial communities that we identified to be enriched at 65% moisture content. Other future research areas involves assessing the impact of moisture content on antimicrobial resistant genes. Also, looking at these enriched microbial communities, can we carry out microbial challenging, whereby you try to treat the swine waste with this microbial community so that when the black soldier fly feeds on them, they are able to effectively break down the organic matter in the waste materials. These are the references and acknowledgements. Thank you to everyone who helped in the Manga Lab, to the North Carolina Biotechnology Center for sponsoring the project, and everyone who is here to listen. Thank you very much.
Thank you. We have time for about 2 questions.
Sorry if I missed this. So what happens to the larva once they process the waste?
So once we process the waste, the larvae, you can harvest it because it contains protein and can be used as animal feed.
Okay, thank you so much for your presentations. For me, I would like to ask about, I was wondering, how will be this project will be broadened and with industrial scale project? Because you said that we can see to the alternative, we also treat these into the wastewater and also, antimicrobial beads, right?
Sorry, can you repeat your question? Sorry.
How can you broaden this project into industrial scale?
Okay, you're saying how to broaden the project into industrial scale. So you have to depend, because the black soldier fly, they can eat large quantities of waste within a short period of time, depending on the larval density. So if you can get as much black soldier fly with your waste material and put them in very suitable conditions to treat the waste material, within a short period of time, they'll help in decomposing the materials.
Okay, so we're going to have the next presenter, and that's going to be Fatema from Indiana University, and she'll be talking about a modified compact dry assay as a rapid alternative for ESBL E. coli quantification. Fatema, the floor is yours.
Okay. Good afternoon, everyone. My name is Fatema Mahua. I'm second-year PhD student at Indiana University, Bloomington.
And my name is Allison Holder. I'm a fourth-year undergraduate student at Indiana University, Bloomington.
So, Today, we'd like to share our method validation study. It's basically evaluating an alternative rapid approach for detecting ESBL E. coli, and we also share some key findings from its field application. So, for ESBL E. coli culture, WHO tricycle protocol recommend a traditional TBX media with cefotaxime, which required media preparation, sterilization, plate pouring, and it's involved a lot of steps. And doing all of these things takes like few hours, and it's very challenging for dealing with environmental samples and routine monitoring of environmental samples. To simplify this process, we explore compact dry assay. Compact dry plates, it's ready to use and commercially available growth medium. It's for faster processing. We can just directly inoculate, directly transfer our samples, and it's has a fewer opportunities for contamination. Also, it's more cost effective compared than our traditional TBX media. Each compact dry plate costs around $0.66, while TBX plate costs $0.90. So, the compact dry plates have several advantages, but it had not been validated yet. So, our goal was to evaluate its accuracy and reliability for detecting ESBL E. coli. So to address the study, our study mainly focused on three research questions. Does compact dry completely accurately detect both ESBL and resistance and susceptible strain? And does the cefotaxime inhibit of their growth on compact dry plates? And are there any significant differences in their growth between TBX plates and compact dry plates? So we use both resistant strain and susceptible strain. We use for resistant strain to E. coli and to for Klebsiella. And in addition, we use susceptible strain. It's as for our control. And we get all of this pure isolate from berry source. For culturing, each strain was revived and regrown on LB agar, and we use 4 microgram per ml cefotaximes for resistant strain to ensure their plasmid retention, but we didn't use any antibiotic for growing susceptible strain. And all of the strains are inoculated in LB broth for enrichment, and then after density checking, we made dilutions and just base dilutions and then plated on both compact type EC plates and TBX plates. And we use a spread plate method for TBX plate, which is recommended by WHO tricycle protocols, but we directly transfer our samples using a pipettes on compact dry EC plates. And we did it 5 times, and we replicated it five times. And for incubation, EC plate incubated at 35 degrees Celsius, and TBX plate incubated at 37 degrees Celsius. And after incubation, colonies were counted, and we did some analysis. So we're moving to the results. Here's some pictorial view of our culture plate. We can see the first image where susceptible strain failed to grow with cefotaxime, and the left second susceptible strain grew without cefotaxime. But on the right side, you can see that a resistant strain grew both in absence of cefotaxime and presence of cefotaxime, this confirming its resistance. And we did independent t-test for seeing the effective cefotaxime on growth for compact dry media. You can see that as expected, susceptible strain, the blue one grew well without cefotaxime, but there is no growth when we added cefotaxime. And in contrast, the orange one, the resistant strain, grew both well with and without presence of cefotaxime. And we can see that there's mean difference, there's no mean difference, and the p-value is 0.482. This study is not statistically significant, so we can say that cefotaxime didn't significantly affect resistant strain growth. And our second plot, that growth comparison between compact dry plates and TBX plates, you can see that their mean difference is quite similar, like 8.78 log 10 CFO for CD plate and 8.886 for TBX plate. And it's also not statistically significant, so we can say that no significant differences between CD and TBX plates. So here's our take-home message. Compact dry can be a reliable and rapid alternative for AMR surveillance. It will take your time, save your time, and also your money.
So I'm going to be talking about the field application for this work in the rapid detection of ESBL and E. coli in environmental samples through the Dig Indy project, which is an evaluation of sanitation infrastructure in Indianapolis. So we're wanting to evaluate the Dig Indy project by monitoring water quality both before and after the tunnel's completion. And so our baseline data is being collected from January 2025 to December 2025. The tunnels will be completed in December of this year, and so our follow-up data will be collected January through December of 2026. So what causes a combined sewer overflow? So during dry or moderately wet weather, sewer from like residential and commercial buildings are able to go to wastewater treatment plants. However, during heavy rain, these sewer systems get overwhelmed and the sewage ends up going to an outfall pipe into a river, and not all of it makes it to the wastewater treatment plant. And so in Indianapolis, Indiana, the Dig Indy tunnel system was created. It is an underground tunnel system that is 200 feet below ground and 28 miles deep, and it's able to store over 250 million gallons of wastewater, and that's what is going to capture the overflow during heavy rains. And this project costs $2 billion US. And so for our methods, this is a quasi-experimental design, like I said, in Indianapolis, Indiana, in USA. It's a 2-year study where we are collecting water samples from 6 different sites over 3 different bodies of water, the White River, Fall Creek, and Pleasant Run Creek. And for each body of water, we are collecting samples from 2 sites, one upstream and one downstream of the CSO affected zones, and we're doing this sampling bi-monthly. And so for our culture methods, so in the field, we use these dialysis filters to filter 120 liters of water, and then we take it back to the lab and create a backflush solution like you see in the middle. And then after this, we create a dilution series, and we plate them on the compact dry plates for E. coli, and then for the ESBL E. coli, we use cefotaxime. And so for some of our results, this figure is showing 2-day rainfall effects on E. coli. So on the x-axis, we have 2-day cumulative rainfall in centimeters, and on the y-axis, we have the log transformed E. coli. And so you can see for each extra 1 centimeter of rainfall, E. coli increases by about 0.37 log units, which is roughly a 2.3-fold increase. And then this figure is showing the 2-day rainfall effects on ESBL E. coli. And so we can see that rainfall is strongly associated with increased ESBL counts, with each centimeter rise in rainfall corresponding to about 0.30 units higher ESBL E. coli levels. So both E. coli and ESBL E. coli are statistically significant. And so our post-intervention data is going to be available by the end of 2026, and the molecular data is going to be available next year. which is targeting enteric pathogen genes, antimicrobial resistance genes, and fecal source tracking markers for comprehensive AMR surveillance in environmental waters. And then if anyone has any questions about these methods, we actually brought compact dry plates, so we'd be more than happy to answer any questions. And then lastly, we would like to thank our PI, Dr. Drew Capone, as well as the other members of the Capone Lab, as well as the funders and collaborators. Thank you.
Thank you so much for your presentation. We'll have time for about 2 questions. Yes.
Hi, great presentation. I'm curious, are you using the bacterial results from this to inform like the state government, or like are they being communicated with policy or anything like that? I'm curious.
No, not right now. We are just doing our like pre-intervention sampling right now. So after December, maybe we'll get more data about this, then I think we can think about that.
Did your slide say the tunnels are 25 miles underground?
No, it's two.
2.5?
It's
Lane? Just
Okay, how deep are underground?
it's 250 feet, I believe, underground.
yeah.
20 miles, 200.
Yes.
I mean, I knew 25 miles was impossible, so I was like, maybe I
Yeah.
yeah.
Any other question? All right. We are looking for you. Okay, let's try to get this done. This one? Okay, we're going to have our last presenter. Okay, we're going to have our last presenter, and she'll be talking about enhanced antimicrobial activity in light-activated silver nanoparticles against superbugs through sequential light exposure. And that would be Anjumana, I think Jannati from UNC Charlotte. The floor is yours.
Hello everyone, welcome to my presentation. I'm Anjumana, a PhD student from UNC Charlotte, and today I'm here to present my work. The title is quite long, but simply say the problem we are targeting here is superbugs, also known as antibiotic-resistant bacteria. And the solution we are proposing is the use of light-activated silver nanoparticle along with sequential light exposure. So I'll be talking about the background of this study, hypothesis and objectives, experimental design, results, and conclusion throughout my presentation. So let's talk about the problem first. Antibacterial resistance, or AR, which is often referred to as silent pandemic, but in reality, it's anything but silent to the millions of people it impacts. It occurs when the bacteria evolve and become invincible to the drugs designed to kill them, making routine infections into life-threatening conditions. It occurs due to several reasons, mainly misuse or overuse of antibiotics. That includes overprescribing of antibiotics, overuse of antibiotics in agriculture, not finishing the doses, poor infection control in hospitals and clinics, also lack of hygiene, and last but not the least, the lack of new antibiotics being developed. So what made things worse is no new class of antibiotics has been discovered in past few decades. So we are basically addressing that field. So if we do not intervene novel approaches, the consequences are catastrophic because projection says that the AR-related infections are about to cause 10 millions of deaths by 2050. So we are kind of like running against the time. So there are some strategies that are available and in use right now, like combination of antibiotics, but those struggles with the development of resistance as well. So we need to find out some solutions or alternative that this bacteria can be surprised by and cannot develop resistance so easily. And silver nanoparticle is such a candidate that has that capacity. So it uses multiple mechanisms to induce the bacterial growth, unlike the conventional antibiotics. But the problem with silver nanoparticle is the main antibacterial effect of silver nanoparticle. It's associated with the release of silver ion, which is a slow process, often takes up to days. But we can overcome this limitation if we can just functionalize or modify the surface of the silver nanoparticle that can provide an oxidative environment, which is very crucial for controlled release of silver ions. So here comes the light-activated silver nanoparticle into the picture. So photosensitizer, which is a light-activated molecule that gets activated by light and produce reactive oxygen species that have the potential, that can provide that required oxidative environment near the silver nanoparticle that is needed for silver ion release, faster silver ion release. So in this work, we used PP9 or protoporphyrin 9 as a photosensitizer, which is already widely studied. So this combination, we are calling it light-activated silver nanoparticle or PP9-AGNP. Under the presence of visible light, it enhances the silver ion release and provide better antibacterial effect. And the advantage of this combination is it efficiently kills resistant bacteria and has low toxicity in mammalian cells, which we have already found and published by our lab. So here, particularly in this research, we have taken this a bit in further level. So our hypothesis was, if we apply multiple irradiation with this PP9-AGNP, that will enhance the silver ion release more than the single irradiation, which will eventually lead to improved and accelerated antibacterial activity compared to the single irradiation. So our objective was to compare the antibacterial activity of PP9-AGNP with single and multiple irradiations, and also to assess the efficacy of PP9-AGNP at lower concentration with multiple irradiations. So here our motivation was to use as less concentration of silver nanoparticle as possible, but without compromising its efficacy. So here the experimental design. So simply saying, we use two different kinds of bacteria in this study. One is gram-positive methicillin-resistant Staphylococcus aureus or MRSA. And another one is gram-negative multi-resistant E. coli or MDR E. coli to see how it's affecting in broad spectrum capacity. So we had two different experimental sets, one control where we haven't added any nanoparticle, and another one is our treatment group. Then we exposed our sample to the light, visible light, once for single irradiation and twice for multiple irradiation. And at last, we used the droplet method to count the surviving bacterial cells. Okay, so this is the results of dark controls. As you can see, As you can see, in the absence of light, both for MRSA and multi-resistant E. coli, none of the combination, nor the individual component PP9, AGNP, and the combination at different concentration, they worked well, which means in the absence of light, it's not significantly working. And for the light controls, these are the controls with the individual components, PP9, the photosensitizer, and silver nanoparticle. The y-axis is showing the log inactivation, and the x-axis is showing the components. So as you can see, for MRSA, PP9 with multiple irradiation showed better results than single irradiation, but till 4 hours. After 4 hours, the efficacy started to decrease, which indicates that PP9 has some sort of limitations, which is lower water solubility. Silver nanoparticle also with multiple irradiation showed better results than single irradiation, which proves that in the presence of light, it's doing something. And for the multiple resistant E. coli, none of them worked so well even in the presence of light. So this is the results of the combination of PP9 and AJNP against MRSA. As you can see here, with single irradiation, that is in the middle, it achieved about 5.12 log activation, but just by applying another event of light after 24 hours, it increased to 6.6, which is about like 1.34 log higher compared to the single irradiation. And even when we reduce the concentration to threefold lower dose, which is 0.5 microgram per ml, we have seen that after 24 hours, there was no significant difference between the results we got after 24 hours with single irradiation at higher dose, which was 1.5 microgram per ml in this study. So, which indicates that just by applying another event of light, we are getting similar inactivation or efficacy even at lower doses. And for multi-drug resistant E. coli, we got interesting results. As you can see, the first and second sets of bars. After 24 hours, both with single and multiple irradiation, we got 100% elimination of multi-drug resistant E. coli. But if you see at the same concentration, and if you see the first set of bar, though we have gotten like similar, same log inactivation, 100% elimination after 24 hours, but if you see on 0 and 4 hour bars, which is in gray and orange color, it's showing that multiple irradiation is doing much better than single irradiation, which means multiple irradiation is doing something. Even at lower dose, at 0.5 microgram per ml, we got significant log inactivation. So since we have seen that both single and multiple irradiation, they are achieving 100% elimination after 24 hours. So we wanted to see how fast it's killing the bacteria with multiple irradiation. So as you can see here, with multiple irradiation, similar log inactivation was achieved 6 hours earlier than single irradiation. And 100% elimination was achieved 4 hours earlier than single irradiation, which supports our hypothesis that multiple irradiation enhanced the silver ion release and made faster silver ion release and in an increased amount that made the faster and effective antimicrobial potency. So in conclusion, we can say that, as I mentioned, that silver nanoparticle and PP9, they both had some limitation in itself. But this combination, they showed a synergistic effect by concealing their individual limitations. And also by applying another event of light, which is sequential multiple irradiation, it enhanced the antibacterial effect for both gram-positive and negative bacteria, that's showing its broad spectrum activity. And also we have seen that even with lower doses, just by applying another event of light, it showed significant bacterial inhibition. So the last key point is multiple light exposure enhance the antibacterial efficacy of PP9 AGNP, allowing effective killing in shorter time as well as at lower doses. So I would like to thank my lab, Dr. Munir's lab, my PI, Dr. Munir, and also this is a collaborative work between our department, our lab, and another lab from chemistry department, Dr. Vivera's lab. And this is NIH-funded work. These are some references. Thank you.
Thank you for the great presentation. We'll get some questions from the audience. Yes, let me start with you.
Yeah, I was just wondering, you said the wavelengths were 400 to 700. Was it just a broadband source? Like, was there some specific?
It's not like specific, it's just the visible light, just like the sunlight. We have a light source that resembles the visible light. It's not anything harmful like UV.
Yeah, have you done any testing to see if certain wavelengths it's more sensitive to certain wavelengths than other wavelengths? Is there an optimal wavelength that would excite the silver?
Sorry, could you please repeat the question?
I'm sorry, I was trying to figure out what wavelength was most effective. This is visible light range, like 300 nanometer, 400 nanometer. Because I know with UV light in certain activation of certain metals is dependent on the wavelength. So I was wondering if you evaluated
No, we haven't tried different wavelengths. It's just like we have a certain light source that was purchased from a company, and that was the range like 400 to 700 that falls under visible light. We haven't tried any other light source. It was kind of like optimized, pre-optimized.
Yeah, I just wanted to make sure. So it's like around 400 nanometer to 700. So it's like sunlight, right?
Yeah,
Ah, okay.
just like the sunlight.
I'm sorry, I came in a little late, but I may have missed it. But do you know the mechanism that's occurring here? Why it's enhancing this?
That's a good question. So we are working on that right now to find out what exactly, like what kind of mechanisms it's using. I'm still working on that and trying to like get some results, which is under process.
Do you have any ideas?
Yes, so from from the literature, what we have learned that silver nanoparticle, it itself can produce silver reactive oxygen species, and it disrupt the cell membrane. And also PP9 itself produces ROS, which is toxic or harmful for the bacterial cells as well. Sorry? I mean, you can say that because silver has been like used from the ancient time. They used to use it for like jewelry, utensils, for the water jars, even doorbells, and they're still in use.
Yeah, that's not going to be the last presenter. I have a lot of time for questions. But if you have questions for even the people presenting already, this is the time to engage with them. All right, seems like there's no question.
Can I just
Thank
take
you so
this?
much. This marks